Mico Leucippe skin cells. A Mico Leucippe is a golden white bare-eared marmoset. They are native to Brazil. The Mico Leucippe is part of the phylum called Chordata. There are many subphylum in this particular phylum, and the Mico Leucippe is part of the vertebrata subphylum, as are humans. This phylum's skin cells are used to prevent organs from drying out. They keep moisture in the body. They can also be used to insulate the body and the organs inside. In humans and other mammals, the skin has three parts: the epidermis, the dermis, and the subcutis or hypodermis. The epidermis forms the surface of the skin. It is made up of several layers of cells called keratinocytes. The dermis lies underneath the epidermis and contains skin appendages, hair follicles, oil glands, and sweat glands. The subcutis contains fat cells and some sweat glands. Epidermal stem cells are responsible for everyday regeneration of the different layers of the epidermis. These stem cells are found in the basal layer of the epidermis. Hair follicle stem cells ensure constant renewal of the hair follicles. They can also regenerate the epidermis and sebaceous glands if these tissues are damaged. Hair follicle stem cells are found throughout the hair follicles. Melanocyte stem cells are responsible for regeneration of melanocytes, a type of pigment cell. They produce the pigment melanin and therefore play an important role in skin and hair follicle pigmentation. It is not yet certain where these stem cells are found in humans. A Mico Leucippe has more hair than human beings, which serves to better protect them in their environment. This image shows a segment of human skin. Some cells found in the skin include keratinocyte, stratum corneum, melanocyte, fibroblast, Merkel cells, and Langerhans cells. This image depicts a basic cell. The vacuole gets rid of skin cells' wastes, and the mitochondria make cell energy from nutrients that pass through the cell membrane or that come into the cell through endocytosis. Skin cells react to external stimuli. For example, after skin has been wounded, inflammatory cells are recruited to the area. First come neutrophils, which clear the wounded area of damaged tissue and infectious particles and signal for the recruitment of macrophages to the wound. Some macrophages, stimulated by growth factors at the wound site, assist in the removal of debris and damaged tissues, while others coordinate the remodeling of tissue at the wound site. Macrophages are essential to produce the signals for dermal repair. Fibroblasts of the surrounding skin move into the wounds during various stages of healing. Following a wound, a fibrin clot is often established if blood vessels were ruptured. Fibroblasts simulate the order of removal of fibrin, replacing the clot with a more stable collagen matrix. Fibroblasts also participate in wound contraction, which involves drawing together edges of the wound and undamaged tissue in order to restore skin integrity. During later stages of wound healing, fibroblasts are involved in reordering these collagen fibers into a more stable structure, integrated with elastin. Growth factors produced by macrophages, keratinocytes, and fibroblasts simulate proliferation of blood vessels into the newly remodeled dermis to restore nutrient and oxygen flow.